then
Okay.
set the rules and you can start from there. So welcome everyone. Today we have Professor Manuel Hermosilla from Johns Hopkins University talking about expected profits and the scientific novelty of innovation. This is our fifth in the series of virtual research seminars in the Center for Management of Healthcare Services. And thank you, Manuel, for your time, and over to you. The way we will work is we'll have 40 minutes of presentation and about 15 to 20 minutes of Q&A. While you are presenting, after the 20th minute, you may want to take a small pause and see if there are questions. And I
Mhm.
will also help you in with the questions that may crop up on the chat window.
Okay, great. Thank you, Chirantan. Thank you for the invitation. Thank you, everyone who still hanging out here in the seminar before the weekend. I appreciate this, and I'm excited to tell you about this paper. It's a paper we've been working on for a long time, but we recently last year were able to access very new and cool data, and you'll see why here. It's a very comprehensive data of the pharmaceutical industry, which I think really pushes the research and the results to a new level. So without further ado, let me get started here. This is the motivation, the main motivation that we have for this paper is the well-known affordability problem in the US. You may hear about this problem based on the poster charts of affordability. These are typically personalized therapies that go for more than 1 million per patient per year. Those are extreme examples, but the affordability problem is actually much deeper. It affects people on a regular basis. This report from the Academy of Sciences says that spending on prescription drugs has been rising dramatically to the point that many individuals have difficulty paying for the drugs that they or their family members need. Okay, so now it's getting to be a real problem for most people, and so that is manifested among other things in the perception that the population has of the industry, which ranks very low in polls that attempt to measure this aspect. So this is one of those polls, the Kaiser Family Foundation, and they're asking about the profits. So are these fair profits? One thing that people say is that the pharmaceutical industry makes too much profits, which is not completely false. Perhaps a bit exaggerated, but the most important thing is, well, are these fair profits? Are they getting these profits at the expense of deviating for what should be their mission, which is to help people improve their health, reduce mortality, and so on and so forth? And maybe many people agree with this statement. 2/3 of the participants agree with the fact that they're perhaps making too much profits. Okay. So one way or the other, this has permeated the political spectrum, and we find ourselves in this rare situation of bipartisan consensus that drug prices should be lowered. Something should be done to help fix this affordability problem. We heard, the 2016 campaign had a lot of this, and we just heard about it again earlier this year, although it's stalled in Congress so far. But there is a push to reduce or to try to fix the affordability problem that has both parties agreeing on it in principle, at least. Now, one sticking point to this debate is what will happen to future innovation. And I'm showing you here Amy Finkelstein's QJE paper, which is perhaps the typical reference that people use to make this point is that, okay, well, you may lower profits today, but that means that in the future, and this is a Schumpeterian rationale, firms are going to have less incentives to invest on drugs, and that is not going to be good. And this argument is actually taken to the limit by some industry advocates. This is Joseph Bast, 15 years ago. He's saying, well, if you cut the profits, he is referring to drug importation here. what you'll do is that you'll be cutting off the stream of new life-saving therapies. So all these drugs that save people's, maybe we won't be seeing any of them because there's just no profit that incentivizes firms to invest in R&D. So that's a real threat, and that is one of the big arguments that this drug pricing policies have faced historically in the United States. Now, when you present this argument in front of industry observers, this is one of them, Marcia Angel, former editor of the New England Journal, they may not take it that seriously because they think, people like Marcia Angel, that there's very little innovative research going on in the industry. So here's a quote of her saying, most of the company's profits are not derived from new drugs, but rather from Me Too drugs or imitations of drugs already on the market. Okay, the famous Me Too drugs. And so Marcia Angel is not alone in this. You go into the medicine literature and you'll find many papers that, well, most of the papers that attempt to measure drug novelty, they will conclude something along these lines. I'm not going to repeat these quotes, but they're all saying, hey, after carefully evaluating what's happening with pharmaceutical R&D, we are sorry to conclude that there's very little innovative research going on. Okay, and so this is the background for our research. We're gonna take all these elements and ask, well, how elastic is novel pharmaceutical R&D to expected profits? Okay, so if we change expected profits, these dynamic effects that Amy Finkelstein talks about, will we really suffer? Will the innovation of new life-saving drugs really suffer in the future? Okay. That is the broad question that we're going to try to say something about in this paper. And how we're going to do it? Well, we are going to take advantage of Medicare Part D, a big program that increased expected profits for some drugs targeting conditions suffered by the elderly, people who are enrolled in Medicare Part D. So, Parkinson's, Alzheimer's, some cancers, macular edema, etc., not others. And I'll show you how we differentiate this. So this was a big shock. The program was passed in 2003 and went into operation in 2006. And it's been proven by many papers to have really deeply impacted the industry. And I think you'll see it in our results. It's also the case here. And then on top of that, we have a big sample of clinical trial activity that approaches the universe. And as I said before, one of the key components of this sample is that we can measure scientific novelty under the hood, okay? So we can classify the scientific approaches, the pharmacological approaches that these research activities, clinical trials, are testing. So we can see whether the investment response to Medicare Part D was primarily funneled through scientific approaches that were non-novel, so highly validated in the past, or new scientific approaches that have never been used before. Okay, so that is our question. What do we find? And very quickly, I'll tell you. First, we find that when we look at translational novelty, this is the main measure of novelty that's used in this industry. It's basically measuring how many times a scientific approach has been used before. We find that this demand shock entailed by the Part D program did relatively little to increase novel R&D investments. Part D did have a very big impact overall in the industry, but the lion's share of it was on non-novel R&D. Okay, so R&D that used pharmacological approaches that had been extensively tested in the past. Okay, so there is a marked skewness towards non-novel R&D within this investment response to Part D. Okay. We're going to do a little bit on the mechanisms. I'm not going to show you all the results precisely, but what I will show you and what I can tell you is that after we look at a bunch of different explanations for why this is happening, our results primarily support the technological risk hypothesis. This coming down in the end to the idea that, well, the more translationally novel R&D is also the riskier one, the one that's more likely to fail, okay? So even though firms would like to get translationally novel drugs to the market because they pay off, they have a revenue premium, they may be shying away from it because it's more likely to fail. It's more likely that they will not reach the market and not be able to clinch this premium. And of course, this has implications for how you incentivize R&D, pull or push policies. And I'm gonna, if I have time, I'm gonna talk something about this at the end of my slides. The good news of our results is that the higher expected profits do seem to meaningfully incentivize what we call recombinant novelty, okay? So many drugs use simultaneously different pharmacological approaches. This is the standard of care for many complex diseases. Drug cocktails are used for cancer and HIV and so on. These polygenic diseases, biologically complex, can use pharmacological approaches that are trying to get at the disease from multiple angles, okay? And so based on this idea, we see in our data that many drugs use several pharmacological approaches at the same time. And we can compute how they recombine these approaches, okay? And in this case, we do see that there is a very strong reaction to the positive demand shock implied by Part D. So those are the good news that we can share with you. The one caveat here would be that, yes, that will enable a special source of therapeutic value, but it's not gonna bring you sequential innovation positive spillovers, okay? So it's not gonna bring new ideas, you're just gonna be harvesting the old ideas, okay? So that's one thing that we want to keep in mind as we develop or design policy to foster therapeutic breakthroughs. Okay, so that is what I have for introduction. It's been about 10 minutes more. I'm gonna tell you about how I measure novelty, and after that, which is the central piece of my presentation, after that, I'm gonna stop for questions if you have them by then. Okay, all right, so this is the rough agenda that I have. Well, I'm going to tell you very quickly about the literature. I forgot I would be doing that. Measuring novelty, the data, and then our main results, including the mechanistic evidence. Okay. So related literature, there's a lot. I'm going to be very brief about this. Perhaps the most related literature, which is not small, is a literature that looks at the demand pool effects in drug innovation. Do bigger markets encourage more innovation? The answer is yes, but the elasticities, the measurements of these elasticities vary a lot. One important paper in this literature is Bloom, Cahoot, and Sood in 2013 in the Journal of Public Economics. They actually, they do something very similar to us. They look at whether a Part D incentivized investments. Yes, they did as per the results, but they do nothing in regards to novelty. Okay, so we look at novelty, and that's the main difference. Now, the other difference that we have is that we have now a 22-year sample, much longer, and as it turns out, most of the effects unfold towards the latter part of the sample. So I think we're doing a better job in capturing all the long-term impacts of this policy. There's another literature that's related and that looks at the welfare impacts of innovation, and perhaps the main point here is that welfare is not only coming from the novel drugs, it's also coming from the Me Too's, from the generics. And this is an important point, no doubt. Generics, Me Too drugs come in the market and they lower prices. They increase the assortment, the variety, so that may have horizontal differentiation benefits. And we are all we endorse this literature. The point is, we are not going to be able to say much. We're not going to neglect this. We're just primarily interested on this direct policy question of, well, how elastic is novel R&D to market size, okay? So this may be one thing that we don't have much to say, the precise welfare estimates, and I'm actually planning to say something more concrete about this if I have time at the end of the presentation. And then finally, there is a literature on drug novelty. It's a pretty robust literature, but one problem with this literature is that the measurements are quite superficial in the sense that they are based on drug approval, so counts of novel molecular entities approved versus generics. That is the main approach. And so, of course, that obviates everything that happens before the market. And it also, it is a bit coarse because there is a lot of novelty variation even within new molecular entities, which is a type of variation that we're going to be focusing on in this paper. Okay. All right. So that is what I have for literature reviews. So let me just quickly jump into how we measure novelty. And I'm going to do it here aided by this graph that describes the pharmacology of the hepatitis C virus, okay? And so what you see here in this graph is basically the pathways, the main pathways that the virus uses in the cell. Okay, so it enters and then it reproduces and transmit the signals and so on and so forth. I cannot give you the highly precise descriptions of what is happening here, but what I can tell you, and this is the main point that I want to make here, is that when you look at the drugs that target HCV, you'll see that they tend to focus on different aspects of these pathways, okay? So for example, sofosbuvir, Sovaldi, you know, this is the Gilead blockbuster that was approved into the, you know, something like 6 or 7 years ago, and that went for $200K per patient per year when it was released. That specific drug was highly novel. Why? Because it was the first one to use this inhibition approach on this specific biological target, which is the NS5B polymerase protein, okay? And so there were other drugs at the market at that time. They were doing other things. They were trying to take care to deal with HCV in different, with different types of pharmacologies. But then Sovaldi came in and did this new thing. Okay, we're gonna give it the maximal translational novelty score, and it actually worked really well. Okay, so it was kind of like a magic bullet because it really cured hepatitis C. Okay, so of course the next drug that would come, dasubuvir, for example, that does the same thing, that tackles hepatitis C the same way that sofosbuvir did it previously. This drug is not going to be very novel. We're going to give it a lower novelty score because it's already deploying pharmacology, an approach to pharmacology that has been used before. Okay. Now, Harboni. Harboni is another Gilead compound, and what's special about Harboni is that Harboni combines two pharmacological approaches. It uses the same one that sofosbuvir used, so inhibition on NS5B, but it also uses inhibition on NS5A, so a different protein. Okay, so now Harboni is was the first one to combine these two approaches. Okay, so now we can define a recombinant novelty score for Harboni, and since it was the first one to use this combination, this bundle of TPAs, then it's gonna receive the highest novelty, recombinant novelty score. Okay, and so as I said before, combining these approaches is a good thing from a therapeutical point of view because many diseases are complex. They don't have a single pathway, and this is why drug cocktails are so you know, prominent as standard of care for many complex diseases. Roth et al. Describe this or summarize this by saying that magic shotguns, so drugs that, you know, shoot at many different pharmacologies, may be preferred to magic bullets in the case of polygenic disease. Okay.
Manuel, can I ask a question?
Of course.
So on this novelty measure, is this assigned by a third party, or you guys are constructing it by candidate?
Yes, we are constructing it. So let me tell you specifically how I do that. Okay.
Okay, and the other related question is,
Yeah.
as you described these two examples, I'm just thinking that there could be something new to the farm, something new to the market, and something that's new both to the farm and the market, right? So are you
Yes.
able to capture that in the way you are constructing
Yeah.
novelty?
Yes. So yeah, great question. Okay, let me address this question after I tell
Sure,
you a bit
sure.
more on how we construct it, but I do have an answer for that. Okay, so to your first question, well, how do we construct it? Well, this is, what makes our data set special, is that we have, for each drug, we know what is the target-based action. What is the target-based action? Well, it's exactly what I described before. It's this pair that includes the mechanism, so inhibition, okay, and then the targeted entity, okay? So for Sovaldi, the TBA was inhibition of the NS5B polymerase, okay? And so when we look at the data, we have about 7,600 of this. So, we have a very narrow partition, okay? About 30% of the trials, 25% of the compounds have more than one TBA, okay? So for those, we can construct the pattern. So this is to your first question, how do we construct this measure of novelty? We construct it ourselves based on this data. Now, specifically how? Well, we construct it at the trial level, and we say, okay, for a trial I, that is for stage S, so preclinical, phase 1, 2, 3, etc., and the TBA-K, okay? So we're gonna measure the number of compounds that have used the same approach, the same TBA in stages equally advanced or more advanced before this particular trial. We're gonna call that the number of deployments. And then novelty is gonna be just 1/1 plus the number of deployments, okay? So if this TBA was used 0 times before, like it was the case for Sovaldi, then it got novelty equal to 1, highest novelty, and as it starts accumulating deployments, this novelty score starts decreasing. That's it. Okay, so very simple. We have a bunch of robustness on how we measure this, and hopefully we'll have some time to talk about them. Maybe some of you will be curious, but this is the simplest approach that actually captures most of the variation and the kind of like the backbone of the variation that you will get even when you relax this structure to account for other valid concerns, I will say. And so for translational novelty, we measure these previous deployments at the compound level, okay? And for recombinant at the TBA level, and for recombinant novelty, we measure deployments at the TBA bundle level, okay? So it's a pretty standard, simple, intuitive way to construct novelty, we think. Okay. Now you ask, well, is this new to the industry or new to the market? Okay. And so this is actually a very charged question. So one thing that we did, right, is that we say, okay, what is the most important of the two? So we looked up the TBAs that had been validated in the sense that they had reached the market, okay?
Mm-hmm.
And when we parse the investments response according to this metric, we find no difference, okay? And so, the very few TBAs, we find that Medicare Part B made no difference. So in other words, the investment response to Part B did not organize relative to which TBAs had been validated. So reaching the market was not, a meaningful dimension in our analysis. That is what I'm trying to say. But when we organize this novelty according to what's new to the industry, then we get a very, informative set of results. Okay?
Manuel, I think pharma projects gives a novelty measure, no, for compounds in the pipeline, because you are catching this much later in the pipeline once things are at the clinical trial stage.
Well, okay, so that's something that we're gonna use. And perhaps we're thinking about the same thing. So let me quickly show you, these are the distributions of novelty. So, for translational, we get about 13% of, highly novel compounds that never been used before. So these are straight from the lab to the clinic, okay? 30%, this is the bar that I'm showing you. And then the bulk is, located here at the left tail of the distribution. So most research is non-novel by this metric. But when we look at recombinant novelty, of course, it's much easier to generate novelty here. You just simply have to recombine. Previously used approaches, and so we get, about 2/3 of all trials are recombinantly novel. Okay. Now, does this make sense, right? And again, back to your question, like, did we get this from someone? We constructed by ourselves. Can we be sure that this is a meaningful metric? So we conducted two validation analyses. The first one, looking at this regulatory-based novelty, therapeutic novelty designation awards. Okay, that's a long name, but basically regulators here in the US and Europe, they look at, based on clinical trial evidence, they say, okay, well, are you providing some novel source of therapeutic value? Okay? Are you gonna save more lives with your drug? So firms have very strong incentives to get these designations, because they get longer exclusivity periods, and they get a, kind of cozier with the regulator that may reduce risk as well. So anytime they think they can get it, they try to get it. Okay, and so we looked at this, which drugs got this independently determined measures of therapeutic novelty as per the awards given by regulators. Okay, and so this is the regressions that we run after we assemble those measures based on translational novelty. So these are linear probability estimate. Increasing the translational novelty is associated to much more likely regulatory novelty designation awards, and the same thing for recombinant novelty. Okay, and also for products that have more than one TDA. So these are the more, the shotguns that I refer to. Okay, so this is one piece of evidence that says, hey, you guys may not be so lost with the way that you're measuring novelty. Another one, and this is actually coming from some research that has looked at the novelty within patents. Okay, so they measure novelty by looking at the technology codes invoked by patents in the U.S. And then they look at forward citations. And this is a very common sense intuition, I think. More novel patents, or patents that are for more novel technologies, should have more forward citations. Okay, this is the result that they find. And so we can do the same thing in this case. We can, for a number of drugs, so about half of the drugs in our sample, we can find the patents, the main patent, and we have the number of forward citations. And again, here we find that translational novelty, so these drugs that are being newly deployed in the clinic, they are also associated to patents that receive a higher number of forward citations. Okay, so the relationship is also positive for recombinant novelty, but it's less strong. And this is not so surprising, because, well, these are just mixing and matching previously known technologies. Okay, so this is our way to have some confidence that we are not, our measurement of drug novelty is not completely off point. I'm a half an hour in. I'm gonna stop for questions if anyone has them. Otherwise, I'll proceed to our regressions and so on.
Yeah, if anyone has questions, please float it on the chat window or unmute yourself and ask your questions.
Okay, well, apparently we don't seem to have any questions, so let me move on then. Okay, so I told you how I constructed the novelty measure, but I haven't told you about the data, so let me give you a quick rundown of the data. We use a Cortellis data set, which is very comprehensive. If any data set approaches the universe of the known universe of pharmaceutical R&D. It is this data set. And so we need to select a bit of this data set because we need to link each drug to the Medicare Part D demand shock proxy. And we also need to be able to have the TBA information that is not available for every drug. Okay, but after we do that, we are left with still a pretty big sample, 76,000 trials for 36,000 compounds. These trials initiated within a 22-year time period, starting in 1997 all the way to 2018. Okay, so a very long period, which actually you need if you want to evaluate the impact of a policy on pharmaceutical innovation, because things move slow. Okay, so luckily we have that long period. So this is the key part to implement the identification strategy. Well, Medicare Part D. So here I told you very briefly, Medicare Part D was approved while George Bush was president, and so they needed a program to help seniors with their prescription drug costs. And so Medicare Part D came into town. They started, basically they reduced the cost of prescription drugs for Medicare enrollees, which are 65 years and older. The program was very big, and so I think that one early estimate for the first 5 years after the program went into operation in 2006 was something like $1,500 per enrollee per year cost. Okay, so it was a very significant boost to demand. But of course, not for every drug, but for drugs that are used by these Medicare enrollees, okay? And so how do we measure which are these drugs that were more exposed to the shock? Well, we use the medical expenditure panel survey data, which is a very nice data set that tells us what drugs are being consumed by people in a representative sample and what kind of insurance they have, okay? So we can compute for each drug, for each condition, measure at the ICD-9 level, what is the percentage of prescriptions that were given on a year to Medicare enrollees, okay? And so this is what I'm showing you here in this graph. This is the Medicare market share, which is that precise statistic I just described, the share of prescriptions that go to Medicare enrollees. And of course, we're doing this in the pre-period before 2003. And so at the left of this distribution, you will have drugs targeting conditions such as acne or infertility, things that you don't expect the elderly to get. On the right side, you have conditions such as Parkinson's disease, macular edema, and so all these conditions that you do expect the older people to get, okay? So drugs targeting these conditions, those are the ones that we think will be most or will have been most affected by the passing of this policy. Okay, that is how this identification strategy works. In econometric terms, what we'll do is that we'll compute this dependent variable big N, which aggregates the number of clinical trials that start targeting a given disease in a given stage, so phase 1, 2, 3 preclinical, in a given year T, and then within a novelty category, okay? And I'll describe to you what novelty categories are in the next slide or so, okay? And so what is this gonna be a function of? Therapeutic area fixed effects, year fixed effects, stage fixed effects, and novelty category fixed effects. The parameters that we're gonna be interested in are this beta PC. P are periods that we are introducing to take account into of the potentially gradual unfolding of the policy. And then these parameters are going to be associated to the interaction of this Medicare market shares times the time indicators, okay? And so if these parameters are positive, they're going to be telling us that trials for drugs targeting conditions with higher Medicare orientation had a relative increase in their frequency in a given time period, okay? So if we find that beta parameters are positive, then that's gonna be evidence that the program increased investment levels. Okay, so let's see the results. Well,
Manuel,
yes.
there's a question from Ankit Shah. He asks,
Okay.
does this construct considers relative complexity of the disease being targeted by the novel molecules? I mean, targeting cancer, multiple sclerosis, or genetic disease can be difficult in comparison to infection.
Yes, yes, great question. So this baseline regression, you see, it's including therapeutic area fixed effects. And so there are differences across therapeutic areas. This, of course, is hiding a lot of variability, okay, at the disease level. So cancers are very different. Even within the cancer's therapeutic area, different cancers can be more complex. Some cancers may be more complex than others. So this regression and the construct itself do not directly account for these biological differences that you're measuring. More generally, our measure is disease blind. Okay, so we are computing this previous deployments not at the disease level, but at the but overall. Okay, so that would be the first part of the first part of the answer to your question. But empirically, what we're gonna do to kind of like be sure that our results are not being driven by these unobserved differences across diseases is that instead of estimating this negative binomial model, which is not very amenable to fixed effects saturation, we're gonna estimate an analog fixed effect regression with fixed effects defined at the disease level, okay? And so this is where we're gonna be able to explicitly account for these differences of targeted diseases. You know, breast cancer being much more complex than other type of cancer and so on, okay? So in summary, our measure does not incorporate directly differences across diseases, but some of our robustness tests do incorporate it in the regression. Okay? All right. Okay, and so this is the categorical partition that we implement. We are going to have 4 categories. These are translational novelty categories, categories 1 through 4. Okay, and so what we're going to do to generate these categories is that we're going to take the distribution in the pre-period, so before 2003. And then we're going to say, okay, you know, these trials that are using a TBA for the first time, okay, novelty scores of 1, we're going to put them in one bucket, the most novel category, T4. And then the other trials, we're going to split evenly, okay? So terciles. This is using pre-data only. And then for the rest of the sample, the post-period, we are going to assign trials based on these thresholds of the pre-period distribution, okay? So this threshold here between T1 and T2 was 0.59 based on the pre-data. Then trials that happened after 2003, we're going to assign 2 categories based on this set of thresholds here, okay? And so you see here, and this actually gives out some of our results, that when you look at the full sample of trials, they start to accumulate in the least novel category, T1. That's already giving out part of that part of our result. Not all of our results, because we are interested in the difference between Medicare-oriented and less Medicare-oriented drugs. This is what we're showing here. The dashed lines here are showing the drugs targeting diseases with above median Medicare, okay? And the dotted and the solid lines with below median, okay? So these dashed lines are the trials for the Parkinson diseases, for the cancers, for all those drugs that are more likely to be suffered, that target conditions more likely to be suffered by the Medicare people who now have higher demand. And so if you look at this T1 category, okay, you see that there's it was parallel before the policy, roughly parallel. The parallel trend tests do not show any red flag. I won't show them to you here. But then eventually, years after the programs became into operation, like 4 or 5 years, there is a clear separation, okay, for the least novel drugs. Something similar, although less pronounced, happens for the next least novel set of trials, T2. You also observe some separation, but it's harder to tell. But then when you move to the most novel categories, T3 and T4, now there is a very little difference, at least not perceptible in a graphical way, okay? So this graph summarizes our main result. The next set of results basically formalize it with our model. You see that the effects, these are the betas, the interaction betas that I showed you before. They're concentrated largely in the first two categories, the least novel ones. There are some impacts on the most novel ones, T3 and T4, but they're minor, relatively speaking, and they unfold much later, okay? So now to get a sense of how important these impacts were, I'm showing you here simulations for the counterfactual scenario in which there is no Medicare Part D, okay? And I'm only considering two categories just to make the picture simpler, T4 and T1, okay? T4, this is the most novel one. It's in gray at the bottom, okay? So without Part D, this is the dashed line, you would have had something that's very similar to what you had with Part D. For the least novel category, it's very different, okay? So the bulk of the investment response to the Part D shock here seems to have been funneled through relatively less novel R&D in the translational sense. If we focus on the 2012-2018 period, the impact for T1 is 80%. Part D is attributed to 80% more trials. T2, 40%, T3, 22%, and T4, 20%. Okay, so the very marked tilt towards less novel investment. Okay, that's our main result. And translational novelty, I told you before, this is the metric that most of the studies, that all of the studies, I will say, that look at novelty in drug development have focused on, and this is why we kind of like spend most of our time here. In the interest of time, I'm not gonna tell you about all these robustness tests that we did to make sure that our assumptions for the quantification of novelty were not driving the results. But you see here, these are all different specifications. Let's measure novelty in this other way, okay? Or categorize it in this other way, and so on. And so what you see here is that the correlations with the baseline scores, with the scores that I showed you, are generally high. And even in the worst case scenario, which is in these 5 and 6 cases, the results, when you rerun the regressions, are very similar to what I just showed you, even stronger for the point that we've been making, okay? So this gives us some confidence that this translational novelty skewness of the investment response is not, it's a rather robust result in which we promote as the main result of the paper. Very quickly, I'll tell you about recombinant novelty. I don't have much time left, but I think I can tell you, I have some minutes to tell you. Here, we also separated categories, drugs in terms of their scores in categories. R1 is just all the bundles that have been used before. Pretty sharp change. Impact of Medicare Part D. Category R2, these are all the trials that are pure recombinations, in the sense that they're recombining previous TBA bundles, but all TBAs that have been used before. So there is no translational novelty here. This is all pure recombination. And again, in this case, there is a pretty significant change. I anticipated this before. Category R3, these are also new recombinations, but they use at least one TBA that has never been used before. So there is a translational novelty component to it. And accordingly, here you don't see much change. You could have predicted this from our previous results, okay? So the main result here is that you do see a pretty significant change here. And these are good news in the sense that these new market rewards that were established by Part D, they are incentivizing this type of novelty, okay? And so that's good because it's good for cancer and so on. But of course, there is not new science that can be used by innovation to come. So there are no sequential innovation spillovers associated to it. Okay, all right. So I'm gonna skip these numbers because you got the point. I am 45 minutes in total. So let me quickly tell you about the mechanisms that we considered. I'm only gonna show you one set of results, but what we want to explain here is, well, why do we observe this tilt towards investments that are, that have the least amount of translational novelty? Why are firms so inclined to focus on these TBAs that have been used before so many times, as opposed to going for the more novel TBAs? Okay, and there are a series of explanations. If you can think of other one that I'm not listed here, I'd love to hear them. So by all means, let me know. But these are the explanations, the potential explanations that we consider. One of them is that, well, maybe, these translationally novel drugs do not make more profits. Maybe actually it's the opposite. A new drug comes into the market, people don't know what they're doing. There may be some skepticism, and so they make less money. So that would be a revenue penalty, okay? So we looked at this. We used some data from top sellers in the US, and we actually found that translationally novel drugs tend to make more money than less translationally novel drugs, okay? So instead of the revenue penalty, they have a revenue premium, okay? So that is the first mechanism that we, that our evidence is not consistent with. So it's not that translational novelty doesn't pay off. Okay, it doesn't appear to be that. So now, based on this result, you can say, well, maybe, this premium is so large that every opportunity for translational novelty has been invested on. Okay, so maybe even before Part D, any kind of like novel basic science that we could get our hands on, we move directly into the clinic. So it's an issue. It may be an issue that basic science is not giving us a supply of translationally novel candidates that we can invest on. Okay, we are maxing out. We were maxing out before Part D, and we are maxing out after Part D. Okay, because it's profitable. Okay, and so what we did to look at this is that we separated the investments in our data set in terms of how early stage they are. We have preclinical trials, happen before clinical trials in humans. These are clinical trials that are largely run on animal models, okay? And then on the other hand, clinical trials on humans. And so what we saw in the preclinical trials is that the investment response to Part D was much more homogeneous. So in other words, there was a pretty robust investment on the most translationally novel categories, okay? So the way that we interpreted that result is that, well, if we're seeing this response at early stage happening not so long after the program was enacted, then there must have been, a large enough or a rich enough supply of translationally novel candidates for this to have taken place. Okay, so this is the evidence that we're showing to address this second hypothesis. Okay, we find that early stage there was a translationally novel response, and that is inconsistent with a limited upstream supply argument. Organizational scope, okay? This is a fairly a prominent argument in the industry, it being that small firms, small biotech firms, are more prone to generate novel candidates, okay? And why is this? Well, because these firms are in closer contact with academia. Many of them are funded by professors. And so these guys are at the frontier, scientific frontier. They're more likely to generate the novel candidates, the translationally novel candidates. But at the same time, because these firms may be more mission oriented or have less resources, they may not have been able to react to it, to the Part D shock. Okay, so it would be the combination of these two elements, the innovation residing on these firms and these firms not being able to react to the Part D shock, that would kind of give us a milder translationally novel response. Okay, and so when we look at the data, what we do is that we separate the responses between big and small firms, firms that are not established, firms that don't have compounds in the market. Those are that is our definition for small firms. And what we see is that this non-novel translational tilt was actually present in both types of firms. So the small firms that I was just talking about, they also responded with non-novel trials, okay? And so that is our piece of evidence that addresses this third organizational scope hypothesis, okay? And we think that is not driving our results. The last one is an old argument saying, hey, translation in novel science, this may be more risky, okay? There is an independent literature in medicine that kind of backs up this argument and spells it out. It says why. So these novel mechanisms may be harder to validate. People may have harder times designing the clinical trials. They don't know who to exclude and so on because they don't really know the science. And all this may cause these trials, may give these trials a higher failure likelihood, okay? And so that's one thing that we could test directly. We know what is the outcome of trials. Do they fail in the sense of does the process stop there or do they keep going? That would be a success. And here we are regressing an indicator of stage-specific success, the alternative being failure, on our measures of novelty, translational novelty, which is what we're interested in. Okay, so columns 1 and 2, they regress on the continuous score and the categorical scores. The difference with columns 3 and 4 is, and in columns 3 and 4, we are using disease-specific fixed effects to account for that heterogeneity that the therapeutic area fixed effects do not account for. Okay, and so as you'll see, more novel drugs are less likely to succeed at stages, and the effect is monotonic and very robust. So this is what we are taking as an argument to say, hey, probably the reason why we are observing this result is risk, okay? And so very quickly to conclude, what do we take out from this? Well, if you want to incentivize therapeutic breakthroughs, pull or push, pull being, tweaking monetary awards, or push, helping collaborations, reducing risk. For translational novelty, it seems that you should incentivize push, and that's actually something that the industry is already doing with these designations. FDA gives you a priority review. That's great. It reduces risk, make things go faster. But the problem that I can identify here is that you need some clinical trial evidence to get this. And it seems that the problem is being generated before clinical trials. Now, the framework that we have, this allows us to identify novelty ex ante before clinical trials. So in theory, it would give us a way to be able to assign these benefits before clinical trial evidence is generated. For recombinant novelty, that's good news. It's great to hear that the market does seem to incentivize them. Pool incentives seem to work for them, but of course, there's no sequential innovation spillovers. Now, how to combine this, that's a difficult question that has to do with, welfare. Does novelty, how much does novelty increase qualities? Okay, it's a big question, very difficult to unpack. We are trying to do something along these lines, but ultimately the piece of information that you need to fine-tune any policy of this sort. Okay, I'm sorry I went longer, but since there were no questions, I felt the confidence. We have a few more minutes in case there are questions I can address.
Yeah, there are there are questions on the chat window, but I just wanted to let you know that this qualis by novelty thing, you probably know this already, that Frank Lichtenberg has a series of papers where
Yes.
he's measuring entry
Yeah.
of NCs and its effects on mortality in cross-country panels.
Yeah.
Those estimates probably can be brought into your work to make some like calculations on welfare.
Yeah. So the challenge there. Yes. Thank you for bringing that up. The one challenge I think of is that, all these qualities that you see are estimated from drugs that reach the market.
Yeah.
And so that is 10% of all drugs that enter clinical trials. So there's a big selection. What you would ideally like to do is kind of like start before entering clinical trials and say, okay, what's my expectation of the qualities that I'm going to generate if I get to the market? And so that this is not a fatal flaw, but it probably imposes a big chunk of selection in terms of.
Yep. Okay, so Mayank has a question which I think you've already touched on a little bit. He asks, is there any impact of basic science research on the trials?
Mm-hmm. Yeah, so it's a big question. One thing that I'll say is that NIH funding throughout the period, and even when you look back like 20 or 30 years, NIH funding across disease categories has not changed much. Relatively, has been increasingly, but it hasn't been overly changing. Now, we need something more precise than that, right? We would to directly test these hypotheses, not relying on this partition that we made, early versus late stage. We would need to see if the funding was going to more novel projects, which is not something that relates directly to distributions across disease categories. It is finer than that. And so that is something that we are interested in, actually, to answer this quality question. Maybe we can get some exogenous variation from there, but at the surface, at least, it does not, things do not appear to have shifted by much.
Okay. Ankit has an interesting question saying that I understand that this construct has used patent citation as one of the factors for novelty evaluation. Can it be used the other way around? I mean, can this construct be an surrogate that patent office can use for checking the novelty parameter for drugs while evaluating patentability? This might help in controlling Me Too innovation.
It's a great question. Yes. Yes. In theory, they should, right? Well, one thing that I will note is that, I've been looking at this timing of when to patent, and many drugs, many firms are playing this game of, well, if I patent earlier, I have less time if I reach the market and so on. And so about half of drugs are patented before clinical trials, about half are patented after, of those that are patented. And so in theory, the patent officer should look into that, right? If the TBA, if this pharmacological approach is essential to the patent, it should be the prior art, okay? And they should be looking at a broader sample than we are looking. We're just looking at anything that gets to the clinic. They are looking at everything that has been patented, okay? But I agree that it's useful information, and my hunch is that they should be accounting for it. Now, whether they account it with the same, systematicity that we do, I don't know yet.
Okay. And you do some specification checks with paired fixed effects, I'm supposing, right?
Pardon? Could you repeat?
Do you pair fixed effects? Because I was, as you were showing those plots, I was thinking, could anything else be driving this move away of these T1 line in your first baseline result that you showed non-parametrically? So I was just thinking of stuff like, could there be some time varying way stages change in terms of ease with which they are allowed to move ahead, or something about the disease that is changing over time by stages and stuff like that. So there are a bunch of other omitted variable possibilities in the specification. I hope you are testing for specifications with some cross fixed effects.
Yes. Well, I'm not sure I fully get the concern, but one thing that I can tell you for sure is that we have year fixed effect. So um, um, we are looking at the secular changes, temporal changes, um, fixed effects by novelty category and year.
So
Those
if you go back to your unit of observation, I think it's SDCT, right?
Yes, disease stage, year, novelty category.
Right. Now, things may be changing by disease and novelty category, by stage and year, which we are not observing as an econometrician, or by disease and stage, etc. Right? There are all kinds of explanations from the market, from the firm, for the technology. So is
Yes,
it possible to run models with those
yes,
alternative?
yeah. So, in previous versions of this paper, to your specific question,
Yeah.
what we did is that we run these models by category. Okay, so a full set of different parameters for each novelty category. Okay, and so we still got this
Got it.
overall pattern, and that would take care of some of what you're saying.
Got it.
Mhm.
Cool. If there are no other questions, I think we can wrap up. Thank you so much, Manuel. I think it's a very cool paper, and especially in today's times, even more important from a public policy implication, I suppose. So thank you for making time for this, and thank you everyone for joining in.
Thank you, Kiranta, and thank you everyone for attending. I appreciate it. Have a good weekend.
You too. You too have a good weekend, buddy. Bye bye.
Okay, bye bye.